Hi, my name is Josh Javor, and I am part of the Bishop Lab at Boston University, and today I'm going to be talking to you about thrust area 1, the nanomanufacturing thrust, where we talk about how to put cellular glue, plan to put cellular glue on some of the nano scale scaffolds that are built in thrust area 2, now it's White Lab. So the overview and goals of this section are to translate highly engineered deposition techniques, atomic calligraphy in our lab, and organic vapor jet printing at Michigan, from fields of physics and chemistry to self-happening scaffolds in the following steps. So first, our first goal is to pattern resolution and deposition speed in 2D and 3D. The second goal is to develop nanoscale patterning control of the cellular glue, and finally, to develop hierarchical multi-material features in 3D. So I'll start with an introduction of atomic calligraphy. There are two main types of atomic calligraphy. We've broken them down into dynamic atomic calligraphy and static atomic calligraphy. So dynamic atomic calligraphy is being worked on by PhD student Lawrence Barrett here in the Bishop Lab, where if you look on the diagram on the right side, the top right, you can see that there is a substrate that is of any arbitrary material really that can go in vacuum. And then you have, just below the substrate, you have some sort of MEMS, some sort of silicon micromachine structure that can move in 7 degrees of freedom. And there's a little hole between the substrate and where you see the evaporation source. And when material is evaporated in the vacuum, it will propagate towards the substrate where it'll pass through that small hole. And when we move that small hole in 7 degrees of freedom, we are able to spray paint on the surface with atomic scale resolution of whatever the evaporation source material was. So this is dynamic because while we are evaporating the source material, we are able to actuate lots of little microscale motors, which you see in the bottom right picture, with comb drives, flexure springs, various other mechanical micromotors in order to move that aperture around and write some prescribed pattern on the surface. What is currently more applicable to CellMet at early stages is static atomic calligraphy. And so static atomic calligraphy is very similar to dynamic, except for we now don't move the MEMS structures while depositing material. And so instead we develop masks. These masks are milled by a gallium ion beam to form whatever patterns, arbitrary patterns that we'd like in a silicon plate, and material is deposited through the plate onto, again, an arbitrary substrate. In this case, of interest to us are any polymers that are made in thrust area 2 with nanoscribe or however, perhaps on parylene or on silicon to do parylene tests. Finally, with static atomic calligraphy, nanoscale resolution is achieved by the alignment pillars that are printed on nanoscribe material, which you can see in the top right. You can see 4 posts, and then if you look at the bottom figure, you can see that there are corresponding holes in the silicon mask that align on top of those features, and so this gives us repeatability to deposit on top of that material. So the atomic calligraphy technology is really a realization of an idea that dates almost as far back as Richard Feynman, where in 1959 he gave a famous speech titled "There is Plenty of Room at the Bottom," where he was describing ideas to use large machines to make smaller machines, which could then make even smaller machines. And atomic calligraphy is effectively a realization of that idea. Atomic calligraphy is a nanoscale 3D printing technique that boasts an ability to write such high-resolution patterns in millimeter squared areas. Such a technology will enable the manufacturability of new classes of metal materials, like optical cloaking, high-resolution light microscopy, and nanoscale patterned cell scaffolds. When it comes to CellMet, many microscale features of cells, such as cell shape and extracellular matrix patterning, have been investigated for their effect on cell function. Tools for discovering such relationships at the nanoscale have been limited and difficult, requiring interdisciplinary collaboration. And that's where CellMet comes in, using the technology of atomic calligraphy to paint such nanoscale patterns. Some challenges for atomic calligraphy are the effects of line of sight deposition and the desire of customization at large larger scales. I describe these as challenges because these are some of these things we can get around by playing tricks in the fabrication process. And I'll show you that in a second with some results that we've already got. But here, what I mean by line of sight is that the surface must be exposed. So the material is really following a line of sight, a straight pattern from, or a straight trajectory from the source, and is landing on the source and being deposited. If you were to deposit at angles through an aperture, this affects your resolution in that you can have blurring of fine lines if you're depositing multiple layers. Finally, the surrounding topology affects the resolution, as we saw with the posts and with the dynamic atomic calligraphy, where if there's a surrounding difference in topology, if there's sort of roughness outside of the area that you want to print on, the mask or the aperture can only get so close to the surface, which will cause a blurring of whatever feature you're trying to print on the surface as well. And finally, throughput, which when we're talking about a nanoscale 3D printer is one of the best figures of merit to rate such a thing. So for dynamic atomic calligraphy, it's good for repeated 100 micron squared patterns in a millimeter squared area, and that's once the entire technology is scaled up through Foundry. Right now we print a single 100 micron squared pattern area, and we can do anything dynamically there, and it will. And then finally, dynamic atomic calligraphy is time-consuming for customized patterns beyond 100 microns squared, as we would have to have this. Dynamic atomic calligraphy is time-consuming for customized patterns beyond 100 microns squared. Static atomic calligraphy then is good for large-scale custom patterns and can withstand repeated deposition, as mentioned previously. However, it's time-consuming for consistent customization and alignment. So if new masks are regularly needing build, that's really best for a kind of prototyping of experiments. But if it was something that you needed changing pattern from experiment to experiment, you really just kind of have to have a set of masks, and customization would be a bit time consuming. So the main focus of thrust area 1, as highlighted in the red circles, are atomic and molecular glues, knowledge base, and propagating up to technology base to developing a technique where we can deposit cellular glue really in whatever patterns that we like. Finally, the technology that I've described here will be applied to Summit in the following manner. And the pictures on the right highlight some results already gathered by a postdoc named Jeremy Reeves here in the Bishop Lab. The CellMet ERC is going to apply this nanoscale printing technique to as cell adhesion sites on biocompatible scaffolds printed in thrust area 2. So atomic calligraphy will print metals, particularly gold, to define the resolution of a pattern. And then another technique from Michigan University, organic vapor jet printing, will then deposit this cellular glue on the surface, which will preferentially bind to the atomic patterns. And then shown on the right, on the top right, is a multi-layer scaffold where you can see there are atomic calligraphy patterns in the in-between layers as well as on top. Those are shown by what look like a split ring resonator feature. It looks like a U, I suppose. So those are actually a metal deposit on top of a plastic that is printed in nanoscribe resin. And then in the bottom right, you see a three-dimensional well where there is a metal deposit on the sides of the well as well as the bottom of the well, where you can see that there is some blurring of the feature as you go deeper in the well, but the pattern is written with decent fidelity. Finally, the expectations from the outcomes of this research are developing a high-resolution 3D patterning scaffold, 3D patterning of cellular scaffolds with cellular glue, and once that is done, we hope to discover entirely new science that will be available for studying cell surface interactions at the nanoscale. It's expected that we will learn how to engineer tissue culturing surfaces to generate predictable tissue remodeling and contractile forces. And all of this in turn will contribute to the two fundamental goals of the tissue engineering field, which are pharmacological drug testing and tissue replacement, where the ultimate goal of CellMed is to develop our tissue patch. So, for example, certain cells leading to pattern formation of tissue may be discovered and could be treated locally by pharmaceutical drugs. And scaffolds could also be engineered to mimic natural biological environments in the development of an implantable heart tissue patch.
So this here is a typical MEMS die where the little square inside is full of micro pattern machines, polysilicon, and on top of silicon nitride in several layers. In a typical atomic calligraphy experiment, we would take the die out of that package, we would put it in this probe station, and in this probe station we have micromanipulators with which we can move around with just barely sub-micron precision, about hundreds of nanometers, and we can move things around, kind of poke things on the surface of that MEMS die. We can move around the really small silicon structures, poke holes in things if we need to, and basically we'll use this setup here to take the atomic calligraphy mask that you saw in the static atomic calligraphy slide, and we will put that on top of the pillars of the nanoscribe print. And so this is where we would align such a thing, and then we'll take the MEMS die from the probe station and we'll put it inside of this vacuum chamber. And so this vacuum chamber is pumped down to ultra high vacuum by a cryogenic pump right there. And then inside the vacuum chamber, we actually have a evaporation source with whatever material that you'd like to evaporate onto the surface, whatever material you'd like to write with. In this case, we're talking that mainly gold is something that we'd be interested in using because it's biocompatible and because you could attach the cellular glue to the gold, which is something like an alkane tile. So inside this chamber, you can't see what's in here, and unfortunately I can't open it up because it's currently under vacuum right now, so the experiment is underway. But inside here there's a crucible, as is shown down here, and inside the crucible, pellets of material are placed inside there. And then inside the vacuum chamber, there is an electromagnetic coil which will be charged with really high currents so that an electron beam will hit the crucible and will heat up the metal. It will raster through the crucible and will heat up the metal such that inside the vacuum, the material will physically puff out of the crucible and then will land on the surface above. So the material is physically evaporating and land on the crucible up here, and it will go through some aperture and land in a pre-prescribed area on the surface of that substrate that we want it to land on. And in order to track how much material is landing on the surface, we have a mass sensor somewhere else inside the chamber that is detecting how much material is evaporating in a given time in a given evaporation procedure. And so in a dynamic or static atomic calligraphy experiment, the only thing that we change is the MEMS that are in between the evaporation source and the substrate. And those MEMS, again, have the apertures on them. So it's whether the aperture has a hole that we move around, much like just writing with a pen, or whether that aperture is just a mask that we can kind of stamp things around with on a surface or something like that. So that's mainly how atomic calligraphy works. It really has its roots in a typical physical vapor deposition technique, and the large difference here to remember is that we put MEMS in between our evaporation source and our substrate. So that's atomic calligraphy.